ही लोग आएंगे, हैं बाबा? सिर्फ इतने बच्चों के साथ करना है बाबा? चलो। Hi, good evening, everyone. This is Kanika Gupta, your TA for this course, and we'll be starting with the session. Just a moment. Yes. So, yes, I hope you have listened all the videos that was uploaded on NPTEL regarding the week 5. So, let's just go through it once. Let's have a very quick revision and then we'll proceed with the questions. Okay. If anyone wants to ask me something in between the lecture, they can. They just raise their hand or unmute your mic and ask me the question. Otherwise, if you want to stay, you want to wait till the end of this discussion, then you can do that also. This is totally up to you. So I'll be starting with the this thing, the microcirculation and the lymphatic system. Basically, at any moment, only 5% of the circulatory blood is in the capillaries that we have studied in the previous week also. That maximum blood stays at any point of time in the veins of the body, not in the capillaries or arteries. Okay, so at any point of time, only 5% of the circulatory blood is in the capillaries. But this is most important. Why it is important? Because it is the only pool from which oxygen and nutrients can be delivered, because while the blood is in capillaries, it is coming through arteries, it has oxygen and nutrients, and when it enters the veins, then it is deoxygenated and the nutrients, maximum of the nutrients has been absorbed by the cells through which these capillaries passes. So yes, these 5% of the circulating blood is important for to meet the oxygen and nutrient requirements of the body. Okay, and when this blood that is transferring oxygen and nutrients to the cells, the waste product from the sorry, the waste product and the carbon dioxide from the cells is being delivered to this blood, which makes this blood deoxygenated and nutrients less. Okay, so the walls of the capillaries are extremely thin. We have studied already that they are made up of a single layer of the endothelial cells, and this constructed of single layer, as I told you, highly permeable endothelial cells. Therefore, water, cell nutrients, and cell excreta can all interchange quickly and easily between the tissue and the circulating blood. Okay, so this is very important that the capillaries should be thin walled and also must have spaces between the cells so that the exchange can be very easy and it the cell can be benefited as much as possible. Okay, So the next thing is exchange of water. How the exchange of water, nutrients, and other substances occur between the blood and the interstitial fluid. Diffusion through the capillary membrane. Okay, capillary membrane, as I told you, it is a single walled tubule, and the cells on the wall, they have some spaces in between them through which this exchange happens. The exchange of fluids and other nutrients, the nutrients which have a larger size. Okay, so by far the most important means by which substances are transferred between the plasma and the interstitial fluid is diffusion. Mostly gases can be exchanged through diffusion, and also there are some transport systems working on the cell membrane through which there is exchange can happen, but other things can be exchanged through this diffusion or these transport systems. So there must be something else there. The figure demonstrates that this process, showing that the blood flows through along the lumen of the capillary. Tremendous number of water molecules are dissolved particles diffuse back and forth through the capillary wall. If you can see these arrows, so you can see that blood or other particles, nutrient particles, are diffusing through the walls of this capillary, and then they are uptaken by the required cell, and also it enters the interstitial fluid through which excess of the fluid enters the lymphatic vessel also. Okay, so the continual mixing between the interstitial fluid and the plasma occurs through this process. Who wants to join? ठीक है जी. So the next thing that happens here is, this is what we are talking. We are starting about the lymphatic system. So this is just the basic knowledge that you guys must have before starting for the lymphatic system. So what Starling hypothesis said, that the fluid moves in the direction of the low pressure. Okay, so if you can focus on this capillary that they have shown below, the direction of the arrows basically. So in capillaries, the capillary that is forming from the arterioles, obviously the blood pressure is a bit higher than than that part of the capillary which is nearing the venules. Okay, so the pressure difference is there. So when the pressure is high inside the capillary, fluid moves outside, because the now this is necessary because the blood here is oxygenated and has nutrients which must be exchanged with the cells and the interstitial fluid so that the cells can uptake it. Okay, so if you can see that arrows are outside, pointing outside, that means the fluid is moving outside. Okay, here the pressure is almost similar between the interstitial fluid and the capillaries, so the direction is in both side. The fluid can go outside and inside both ways. Okay, and when it is getting converted into it is, the blood is getting deoxygenated and the nutrients, maximum of nutrients has moved out. Then if you can see that as the capillary is moving in the direction of the venules, then the fluid is coming inside because the pressure has dropped down as much as that the outside interstitial fluid pressure is higher than the one which is flowing inside this capillary. So the fluid starts entering inside the venue. So this is Starling hypothesis, which shows that the fluid moves in the direction of the low pressure. Okay, so this is how the plasma also behaves, which oozes out of the capillary when the blood is flowing through the capillaries. Okay. Okay, so lipid soluble substances can diffuse directly through the cell membranes of the capillary endothelium. Why it is so? Because we all know that the cell membrane is made up of phospholipid bilayer. So like dissolves like. So phospholipid bilayer will allow lipid soluble molecules to pass through it very easily, while the molecules which are charged cannot pass through this cell membrane easily. Okay, so if a substance is lipid soluble, it can diffuse directly through the cell membrane of the capillary without having to go through the pores. It doesn't require pore because cell membrane is allowing this molecule to pass through this. Okay, such substances include what? As I told you, gases, oxygen and carbon dioxide, because these substances can permeate all areas of the capillary membrane. Their rates of transport through the capillary membranes are many times faster than the Rates for lipid insoluble substances. Lipid insoluble substances generally need some kind of a channel or transport protein to pass through the plasma membrane. They cannot pass as such. Keep this thing in mind always, that a charged molecule or lipid insoluble substance cannot pass through a plasma membrane as such. It requires either some kind of channel or some kind of transport protein to make it pass through that plasma membrane and enter inside a cell or exit the cell. Okay, such as sodium ions and glucose that can only go through either pores or through transport channels. Okay, so this is something very important you should always keep in mind. This is not the case only here. It happens everywhere. Whenever we are talking about a plasma membrane and transport across the membrane, then if the substance is insoluble in lipids or it is charged, it cannot pass through the plasma membrane as such. Okay. Many substances needed by the tissue are soluble in water but cannot pass through the lipid membranes of the endothelial cell. Such substances include water molecules themselves. Water also cannot pass through the plasma membrane as such. Sodium ions, chloride ions, and glucose. If you can see, all of these are not soluble in lipids, so they cannot pass through the plasma membrane as such. तो ये फिर क्या करते हैं? These substances pass through the pores in the endothelium cells. As I told you, the capillary wall has pores between the endothelial cells. Okay, arrangement is as such of the endothelial cells on the wall of the capillary. Okay, so now we are coming to lymphatic system. Lymphatic system represents what? An accessory route through which fluid can flow from interstitial spaces into the blood. Why it is important, we'll see in further slides. But you should know that this system is an accessory route. It is important for the to maintain the plasma RBC balance in the blood. If the fluid that is retained in the interstitial fluids, when this fluid is oozing out through the capillary walls, when it is not returned to the blood, then it can lead to chronic diseases. So lymphatic system is a very important route that our body requires. ठीक है? So most important, the lymphatics can carry proteins and large particulate matter away from the tissue spaces, neither of which can be removed by absorption directly into the blood capillaries. As I told you, large molecules हैं, proteins और जितने भी particulate matter है, जो large particulate matter हैं या proteins हैं, they cannot pass through plasma membrane as such in the capillary. So what happens is, this lymphatic system comes to the rescue and it takes those proteins or large particulate matter back to the blood. Okay, it returns them back to the blood to maintain its homeostasis. Okay, this return of proteins to blood from interstitial spaces is an essential function. Okay, without which we would die within about 24 hours. If this lymphatic system doesn't work properly, then it can lead to serious issues. And if this lymphatic system doesn't exist in a organism's body, in a complex organism's body, then it can lead to the, generally when we are talking about humans, then it can lead to the death within 24 hours. Okay, so yes, this is the lymphatic vessel. If you can see, one thing that you should notice is these lymphatic vessels has a dead end. It is not like they are continuously in connection with one another tube. They have one end closed. Okay, these ends are closed. If you can see, these are the lymphatic vessels, and these are permeable cells through which the fluid flows from what? From interstitial fluid into these lymphatic vessels. Also, these vessels, the endothelial cells that are making the walls of these vessels, they are bound to each other very loosely. They have large spaces in between them, so that large molecule can enter inside the lymphatic vessels. Okay, so this is just to show you the arrangement. Okay, so all of the fluid and proteins that normally accumulate in interstitial compartment as a consequence of capillary filtration are efficiently removed and carried back to bloodstream via lymphatic system. This is what we have read so far. So this is what they have written. It is estimated that for a 70 kg man, The lymphatics return nearly 3 liter of fluid, approximately 120 gram of proteins. You should keep in mind these figures. These are important figures. So you should keep this thing in mind, that approximately 120 gram of proteins to the bloodstream every 24 hours. These figures are generally for 24 hours. These calculations are for 24 hours. Okay, because the recycling of plasma protein is essential for maintenance of normal oncotic pressure. Oncotic pressure we'll see further in the slides, further in the presentation, but you should keep in mind that this is very essential, that the recycling of plasma protein happens to maintain the normal oncotic pressure gradient between plasma and interstitial fluid. Any injury or blockage of the main lymphatic channels may be life threatening. So it is a very important system we have. Okay. Yeah, they have explained here also, oncotic pressure or colloid osmotic pressure भी हम इसको कहते हैं, is a form of osmotic pressure exerted by proteins, notably albumin. Albumin is a large protein which is present in our blood, in a blood vessel's plasma, that usually tends to pull water into the circulatory system. Why water into the circulatory system? Because when these large proteins are present in some kind of a fluid, then it increases the osmotic pressure of that fluid. And we all know that any water has a tendency to move from a higher to move to move sorry it has a tendency to move in the direction which has the higher osmotic pressure to balance out that pressure. So yes, it generally moves in the circulatory system. It is an opposing force to capillary filtration. It generally opposes the capillary filtration, because if the capillary keeps oozing out the plasma, then the concentration of the blood will increase so high, then it will lead to further chronic problems. Okay, so it is an opposing force to capillary filtration to maintain that balance of fluid oozing out through the capillaries and interstitial colloid osmotic pressure. So this is very important factor that you should keep in mind. This is the structure schematic diagram of lymphatic vascular tree. As I told you, lymphatic capillaries are blind-ended at one end. They are attached to other larger lymphatic vessels, while at one end they are just blind-ended. Okay, capillary lymphatic endothelial cells are only partially covered by basement membrane. There is not any phenomena that they are wholly covered by basement membrane, but they are partially covered by basement membrane. Button structures located at the capillaries cells, endothelial cells, and LEC anchor to the elastic components of extracellular matrix by anchoring filament. These are the anchoring filaments. Okay, elastic fibers, and these are the anchoring filament. I'll just open the pointer. These are the anchoring filaments, and these are the elastic fibers to keep these things in place. Okay, and if you can see, as the vessel is the size of the vessel is increasing, the lymphatic vessel is increasing, the smooth muscles around these vessels is also increasing, which is also important for these vessels, because after collecting all the maximum of the fluid, they must supply this fluid to the blood system, and to make this supply they have to move against the gravity also. So they need this smooth muscles to. Kishori, I have admitted you. I hope you are able to join. Yes, she has joined. Sharadinduji, if I could request you, can you please switch off your camera? Okay, everyone. So where we were? Yes, we were talking about this. Yes, this facilitate interstitial fluid and cellular entry into the lymphatic capillaries. Interstitial fluids and cells can enter the lymphatic capillary through both paracellular and transcellular routes. These are very permeable cells, so cells can also interstitial fluid जो है इन cells में enter कर सकता है either through transcellular, मतलब सेल को पास करके भी आ सकता है, और पैरासेलुलर मींस जो स्पेसेस होते हैं सेल्स के बीच में उनके थ्रू भी पास करके आ सकता है. Okay, lymphatic capillaries converge into precollectors which also have incomplete basement membrane. Basement membrane is incomplete here. Okay, and they also have walls to support their single direction movement. As I told you, they have to move against the gravity, so for that they require walls so that fluid doesn't bounce back or doesn't come down again due to the pull of the gravity. So for to support that flow they have walls also. Okay, to support the unidirectional flow. Okay, so the principal deriving force for the entry of interstitial fluid into the terminal lymphatics is what? Pressure of the interstitial fluid. The pressure in the interstitial fluid will be higher compared to the one which is present in the lymphatic vessel, this terminal lymphatic vessels. So obviously the fluid will flow from high pressure to low pressure. So here it happens the same way. As the fluid is filtered from the capillaries, accumulates in the interstitium, which increases the pressure of the interstitial fluid, and this increase in pressure thereby creates an hydrostatic pressure gradient between the interstitial compartment and the lymphatic vessel lumen, and this difference in pressure makes this fluid to flow into these lymphatic vessels. So expansion of interstitium, sorry, also causes the anchoring filaments to pull open the terminal lymphatics. How they pull open it? As I told you, they have spaces in between the cells. So when they are stretched, when the elastic filaments through which these are anchoring to these elastic things, the elastic proteins, when they are activated they just pull open the lymphatic vessels, and this allows the elevated PIF to readily propel the interstitial fluids into the lymphatic vessels. So this is a stretch mediated active contraction of lymphatics which favors, due to this sudden increase in the fluid amount in the lymphatic vessel causes the activates the contraction of lymphatic vessels, which is done by the smooth muscles lining these lymphatic vessels, which favors the propulsion of lymph in the in the forward direction so that it can be poured back into the blood flow. Okay, so when a collecting lymphatic becomes distended with interstitial fluid, a myogenic response. Myo word is generally used for muscles. Okay, so myogenic response is elicited that causes the lymphatic smooth muscles to automatically contract. This causes what? Lymphatic smooth muscles to automatically contract, and this leads to the flow of this interstitial fluid, or we can see lymph here because now the fluid is in the lymphatic vessel to move forward in the direction. Okay. Okay, so the one-way pumping action of collecting lymphatics is the most likely explanation for subatmospheric PIF, that is interstitial fluid pressure they are talking about. It is measured in some tissues because it literally sucks fluid out of the interstitial compartment. Due to this pressure difference, the fluid is actually sucked out from the interstitial compartment and is dumped into the in a lymphatic vessel. Okay, so this is all what we have read so far because lymph vessel. Acha, one more thing as I told you about the walls thing. So क्या होता है कि lymph vessels have walls that prevent backflow. As I told you, it requires the walls so that the blood doesn't move in the direction of the gravity. Okay, so the external intermittent compression phenomena such as, अच्छा, walls तो हैं ही हैं और contraction of lymph vessel is also there, which helps the lymph to move forward in the direction against the gravity. One more thing that is actually important here is it is muscular contraction. When the body is moving, some kind of movement is there. तो जो muscles हैं आसपास की वो भी help करती हैं किसके लिए? To move this fluid further against the gravity and then in the forward direction so that it can reach to blood. Okay, so respiratory movements hogi, muscle contraction hogi, even arterial pulsations also provide some kind of a pressure to move this lymph flow. Okay, so this is what it is. And जब लिम्फ सारा आगे की तरफ फ्लो कर जाता है तो वेयर इट रीचेस टू द वेन्स, ठीक है। लिम्फेटिक वेसल्स जो हैं वो टर्मिनेट होती हैं किसमें, वेन्स में। तो ये सारा का सारा ब्लड जो है ये सप्लाई किसको करती है, वेन्स को करती है, ठीक है। दस द कैपेसिटी ऑफ लिम्फ ड्रेनेज टू इंक्रीज is limited and can be overloaded with high capillary fluid filtration rates. When the capillary filtration rates are high, then sometimes happens is that the lymph vessels are overloaded with the fluid because the amount of fluid that has entered the interstitial spaces is more than the normal one. Okay, but in that case what happens is when this the fluid that is entering the interstitial spaces is much higher than the normal ones, then it leads to a situation called as the edema. Edema is the swelling, we'll see it further. Okay, so this is what happens, this is the edema situation. It happens an increase in interstitial fluid volume is called as edema, or we can also call it as swelling. Okay, इसको swelling भी कहते हैं. Edema forms when the volume of interstitial fluids exceeds the ability of lymphatics to return it to circulation. When the lymph vessels are actually saturated with the lymph flowing through it, then in that situation it is not able to collect all of the fluid from interstitial spaces that it should have collected, which leads to this situation of swelling, or we can call it as edema. ठीक है. It can happen when there is an increased filtration or when lymphatic drainage is impaired. या तो lymphatic system में कोई problem हो या फिर जो filtration हो रहा है capillary से that is high in rate. Okay. Okay. So the protein concentration in interstitial fluid of most tissue की average कितनी है, 2 grams per deciliter. ठीक है. And the protein concentration of lymph flowing through this tissue is near this value only. Okay, so ये इसका कहने का मतलब ये है कि दे ट्राई कि जितना भी प्रोटीन इकट्ठा कर सकते हैं जो कि ब्लड में होना चाहिए था, बट इट इज देयर इन द इंटरस्टिशियल फ्लूइड. सो लिम्फ ट्राइज दैट इट ब्रिंग्स that total amount of protein back to blood. Okay, so the lymph formed in liver has a protein concentration as high as 6 gram per deciliter, and the lymph formed in the intestine has a protein concentration as 3 to 4 grams per deciliter. This is the difference. लिवर में जो लिम्फ बनता है उसका प्रोटीन कंसंट्रेशन होता है 6 ग्राम्स पर डेसीलीटर, और जो लिम्फ बनता है इंटरस्टिशियल फ्लूइड के इकट्ठा करने से उसका कितना होता है कॉन्सेंट्रेशन, 3 to 4 grams per deciliter. Okay. So, because about 2/3 of all lymph normally is derived from liver and intestines, and the thoracic duct lymph, which is a mixture of lymph from all areas of the body, usually has a protein concentration of 3 to 5 because it is a mean. Liver में से ज़्यादा आ रहा है, बाकी body से 3 to 4 आ रहा है, तो it is generally reaching to the range of 3 to 5. अच्छा, जो मेन चीज आप लोग को यहाँ ध्यान देने की है कि सारा लिम्फ जो है एक्चुअली में आखिर में जाके कौन सी लिम्फ डक्ट में पहुंचता है, दैट इज thoracic duct, thoracic lymph duct. ठीक है. This is the thoracic duct in which the lymph is getting collected, from which it is poured into the blood circulatory system. ठीक है. So this is what is a very important thing that you should know. Okay, so if you can understand from this structure, this is what, this is a villi. ठीक है. Sorry, this is a intestinal villus. ठीक है. Villi is used for many, villus is used for single. So this is an small intestine villus, in which if you can see there is this blood supply, there is this extracellular membrane through which the absorption of maximum of things in the small intestine is happening. And if you can see it in the center, this pink structure, this is what the central lacteal. Now what is it here for? This is the central lacteal. This is the central lacteal. What is it here for? It is here to collect the fat which is present in the small intestine, which it got from what? From the food that we have eaten. Okay, so the lymphatic system is also one of the major routes for absorption of nutrients from the gastrointestinal tract, especially fats. ओके. और ये क्या करते हैं, मैंने आज जैसे आपको बताया कि लिम्फ डक्ट्स जो है बेसिकली जाके खुलती हैं किसमें, ये जाके खुलती हैं डायरेक्ट ब्लड में, तो ये सारा का सारा फैट जो है ये सप्लाई होता है कहाँ, टू द ब्लड. Someone wanted to join. No one's here. ठीक है जी. So this is a central lacteal which is important to collect the fats from the small intestine. Okay, let's move to next slide. Okay, elephantiasis. Elephantiasis क्या होता है? It is a condition which occurs due to which nematode? Wucheria bancrofti. ठीक है, this is a nematode and this is what the causal organism of elephantiasis which is transmitted by what? Mosquitoes. होता क्या है इसमें? lymph nodes जो है वो swell कर जाती हैं. ठीक है, the adult worm only live in the human lymphatic system. The parasite infects the lymph nodes and blocks the flow of lymph throughout the body. Sorry. जब ये adult worm जो है ये flow करता है उसमें lymph nodes के through, तो ये क्या करता है? ये उनको ब्लॉक कर देता है, lymph को. वो जो lymph का जो flow है उसको ब्लॉक कर देता है, जिसकी वजह से क्या होता है? chronic edema हो जाता है. A body is prone to infections because ये आपकी lymph nodes को ही infect कर दे रहा है. ठीक है, तो जनरली होता कहाँ है? लोअर पार्ट ऑफ द बॉडी में ज्यादा देखा जाता है इसको. एंड दिस इज द सिचुएशन दे हैव शोन. दिस इज अ एलिफेंटियासिस सिचुएशन. ठीक है, इतनी ये तो बहुत कम है, इससे भी ज्यादा स्वेल कर जाती है कुछ केसेस में. Now let's come to kidneys. These are very important thing and I am hoping कि आप सबने जो है सर की वीडियोस को बहुत ध्यान से देखा होगा, तो I'll try कि मैं इसको थोड़ा फास्ट कवर करूं. किसी को बीच में कुछ और पूछना है या कहीं कंफ्यूजन हो रही है, you can always ask me. Okay, no problem at all. So okay, function of kidney. We all know kidney has a function of filtration to production of urine, so that the excess of these soluble amount of waste and the excess of water can be removed from the body, and it can maintain the regulation of water, inorganic ion balance and acid base balance of the body. ठीक है, ये किडनी का वन ऑफ द मेन फंक्शन है. उसके अलावा क्या फंक्शन है इसके? रिमूवल ऑफ मेटाबॉलिक वेस्ट प्रोडक्ट्स फ्रॉम द ब्लड एंड देयर एक्सक्रीशन इन द यूरिन. जो भी सोल्यूबल वेस्ट जो ब्लड से बनता है वो सारा का सारा क्या करता है ये? It excretes it through urine. And which organ plays the role? Kidney. Okay, removal of foreign chemicals from the blood and their excretion in the urine. ठीक है, ग्लूकोनियोजेनेसिस भी करता है when the situation arrives. We'll see it later. And हार्मोन भी बनाता है. कौन सा हार्मोन बनाता है ये? इराइथ्रोपोएटिन. इराइथ्रोपोएटिन क्या करता है? इराइथ्रोसाइट प्रोडक्शन के काम आता है. What is erythrocyte? It is red blood cells. Red blood cells are also called as erythrocytes. Okay, so उसकी हार्मोन की प्रोडक्शन करता है erythropoietin की. This production happens in kidney. Another hormone that is produced is, we have studied it, we have studied about it before. This is renin. Renin किस लिए जरूरी है? To maintain the blood pressure, which is important for the RAS system. ठीक है, renin angiotensinogen system. And also it forms the 125 dihydroxy vitamin D. ठीक है, which influences calcium balance, basically reabsorption of the calcium. ठीक है, so ये important functions है kidney के, and this last function we'll study it further in the course. Okay, so yes, excretion of metabolic waste products, foreign chemicals and drugs and hormone metabolites. The kidney is a primary means for eliminating these wastes in the urea. By the many, nephrons are involved in this, and there are a hell lot of systems that we'll be studying further, which are involved in absorption and removal of these waste. Either if the compound that is flowing through this filtrate that filters through this Bowman's capsule, it is reabsorbed in the blood system that is flowing along with this nephrons. Or if it is not required by the body, then they will release it in this filtrate to get it removed from the body. Okay, So you should keep this thing in mind, that this is important for the soluble fraction of the waste that our body produces. Okay. These waste products must be eliminated from the body as rapidly as they are produced. So kidney eliminates most of the toxins also, and some foreign substances which are either produced in the body or ingested by the body from outside. Some like pesticides which are generally present on our vegetables. Sometimes the amount is higher, sometimes the amount is lower. Whatever the amount is, it is being removed from the body with the help of the kidneys. Okay. And the drugs that we take sometimes, like some kind of antibiotic we have taken, anything that our body doesn't require anymore, sorry, or they are toxic to the body, are removed from the body through these kidneys. Okay. The urea is formed from the metabolism of amino acids. Creatinine from the muscle creatine. You guys must remember from the lecture. Uric acid from nucleic acids, from the degradation of the nucleic acids. Bilirubin is the end product of which protein? This is hemoglobin, and hemoglobin कहाँ present होता है? We have, everyone knows that it is present in the RBCs, and it is important for the transport of oxygen. ठीक है? The metabolites of various hormones and drugs is also present in this filtrate. Okay. Someone wants to join, or someone has raised their hands. Admit. चलिए जी, ठीक है. सो द रेगुलेशन ऑफ एसिड बेस बैलेंस, या फिर रेगुलेशन ऑफ इरेथ्रोसाइट प्रोडक्शन इरेथ्रोपोइटिन की वजह से होता है. विटामिन D3 क्या होता है? ऑल्सो नोन एज कैल्सिट्रायोल. इट इज प्रोड्यूस्ड इन द सेल्स ऑफ PCT, proximal convoluted tubule of the nephron. क्या होती है proximal convoluted tubule? We'll see further in the lecture. It is essential for normal calcium deposition in the bones and calcium reabsorption by the gastrointestinal tract. So calcium reabsorption के लिए these are very important. And in some situations glucose synthesis also happens in kidneys, that we will study further in the course. Okay. Let's come to the kidneys now. So the structure of kidney, I'll just go through it once very fast. It is, this structure of the kidney is, this is outer part is renal cortex, and this is renal medulla. ठीक है, this is renal medulla and this is renal cortex. This outer part is renal cortex, this is medulla. ठीक है. And in this medulla, if you can see these this structures, these are actually bunch of nephrons. ठीक है. Nephrons is the functional unit of kidney. There are millions of nephrons present in the human kidney, including both the kidneys. ठीक है. This is what renal pelvis, from in which the waste from each and every nephrons get collected, and then this is the ureter, which is the tube through which this filtrate is passed to urinary bladder to be removed out from the body. Okay, so this is the basic structure of kidney. This is the structure of a nephron. What is this? One, this structure is glomerulus. Just, this is glomerulus, or we can say glomerular capillaries. The structure that is covering it is a cup shaped structure. So इसको हम Bowman's capsule भी कहते हैं. ठीक है. The Bowman's capsule. This is proximal convoluted tubule. Proximal means near. So this is near to this Bowman's capsule and glomerulus. So this is called as the proximal convoluted tubule. ठीक है. उसके बाद this is what? This is proximal straight tubule. This is not convoluted. If you can see, this is convoluted. This is straight tubule. Thin descending loop of Henle. This is ascending loop of Henle. And this is what? This 6 is thick ascending limb. And then फिर उसके बाद क्या आ जाता है हमारा? 7th is macula densa. Macula densa क्या होता है? We'll see further in the lecture. Then this 8th is what? Distal convoluted tubule. Okay? This is distal convoluted tubule. Then it reaches to the connecting duct, connecting tubule. and this connecting tubule leads to the collecting duct, and then it further leads to the medullary collecting duct and all these different parts of the collecting duct, and finally to what? The renal pelvis. This is renal pelvis, and from here then this filtrate is removed out from the body through the help of ureter and urinary bladder. Okay, so if you can see, there are two types of the nephrons present here. One is the juxta medullary nephron, and these are the cortical nephrons या superficial nephrons भी हम इनको कह सकते हैं. These juxtamedullary nephrons are deep inside the medulla, while these cortical nephrons are uh, more or less present in the cortex, and some part of them is embedded in the medulla. Okay, so on the basis of that, there's a differentiation of the nephrons. Okay, blood flows to the two kidneys in a normal about 22% of the cardiac output. That is only 22% of the blood that is flowing in the cardiac output. That is what passed through the kidneys. That is nearly 1100 ml पर मिनट. हर मिनट की है, क्योंकि एक मिनट में हमें पता है कि कितना ब्लड फ्लो करता है हमारे हार्ट में से. नियरली 5 लीटर्स. तो अगर उसका आप 22% निकालेंगे तो इट इज अराउंड 1100 ml. ठीक है. So the renal artery enters the kidney through the hilum and then branches progressively. जिससे क्या बनता है? ग्लोमेरुलर कैपिलरीज बनती हैं, जहाँ पे फिर क्या होता है? फिल्ट्रेशन होता है यूरिन का. जो फर्स्ट स्टेप है फिल्ट्रेट का बनना, वो बनता है. देन द डिस्टल एंड ऑफ द कैपिलरीज ऑफ ईच ग्लोमेरुलस कोएलेसेस टू फॉर्म एफरेंट आर्टीरियोल. जब वो आती है तो उसको कहते हैं एफरेंट आर्टीरियोल, ए से शुरू होता है. जब वो लीव करती है तो उसको कहते हैं इफरेंट आर्टीरियोल, ई से शुरू होता है. ठीक है, तो ये दोनों नामों में कंफ्यूज नहीं होना. विच लीड्स टू अ सेकेंडरी कैपिलरी नेटवर्क, जिसको हम क्या कहते हैं? Peritubular capillaries. These are peritubular capillaries. Okay, so it is called as the peritubular capillaries, and the peritubular capillaries then are surrounded by renal tubules. वहाँ पे जहाँ पे क्या होता है? Reabsorption and absorption वगैरह सब होता है. ठीक है, so these things are of very importance. देखिए यहाँ पे if you can see glomerulus, Bowman's capsule, efferent arteriole and all of that. ठीक है. अच्छा, एक चीज और आप लोग को पता होनी चाहिए कि there is when the blood is entering inside this nephron to form the glomerular capillaries, the pressure of the blood inside the capillaries is 60 mm of Hg, and when it leaves through the efferent arteriole, the pressure is 59 mm of Hg. Okay, so there is a drop of 1 mm of Hg. ठीक है. और जो ये पेरिट्यूबुलर कैपिलरीज होती हैं, इसमें जो ब्लड प्रेशर होता है दैट इज अराउंड 13 mm of Hg. ओके, सो दिस इज इम्पोर्टेंट. चलिए जी, कोई आया है, वो कह रहा है. ठीक है, तो ये होता है. उसके बाद नेक्स्ट आता है हमारा ग्लोमेरुलस. ये मैंने आपको बता दिया, इज अ network of capillaries, जो कि when the artery is entering into the kidney splits into many capillaries, and when the artery is leaving the kidney, then these capillaries together come together again and make this efferent arteriole. So this is what glomerulus is. And when they are passing through these glomerular capillaries, a filtrate is formed depending upon the pressure difference, जिसको हम कहते हैं glomerular filtration rate. उसके बेसिस पे जो फिल्ट्रेट है वो ब्लड से निकल जाता है, एंड इट इज अराउंड 1100 ml पर मिनट. ठीक है, दिस इज समथिंग यू शुड ऑलवेज कीप इन माइंड. अच्छा, एक चीज और, द इनर लेयर ऑफ ग्लोमेरुलर कैप्सूल कंटेन्स हाईली मॉडिफाइड ब्रांचिंग एपिथेलियल सेल्स, जिसको हम क्या कहते हैं? पोडोसाइट्स भी कहते हैं. क्यों कहते हैं पोडोसाइट्स? बिकॉज दे हैव पोड्स इन देम. पोड्स क्या होंगे? फिलामेंटस, ऐसे हाथ जैसे स्ट्रक्चर होंगे जो कि ऐसे एकदम बाउंड करके रखते हैं जो कैपिलरीज होती हैं उसको, टू प्रिवेंट द लॉस ऑफ लार्ज प्रोटीन्स. ठीक है, इट इज एक्चुअली इंपॉर्टेंट टू प्रिवेंट द लॉस ऑफ लार्ज प्रोटीन और इंपॉर्टेंट प्रोटीन्स through when this ultrafiltration is happening inside the glomerular capillaries. ठीक है, तो this is important. इसमें लिखा भी है इन्होंने, पोडोसाइट्स टर्मिनेट इनटू फुट प्रोसेसेस व्हिच सराउंड्स द बेसमेंट मेम्ब्रेन ऑफ द ग्लोमेरुलस। ग्लोमेरुलस में जो बेसमेंट मेम्ब्रेन होती है उसके बाहर ही पोडोसाइट्स प्रेजेंट होती हैं, एंड द क्लेफ्ट्स बिटवीन द फुट प्रोसेसेस आर कॉल्ड एज द व्हाट, फिल्ट्रेशन स्लिट्स। The filtration slits are formed by the foot processes of podocytes. You should always keep this in mind. This is where the filtrate enters the capsular space and then that flows through this loop of Henle, proximal convoluted tubule, distal convoluted tubule and every structure. Okay. अच्छा, अब क्या होता है, ग्लोमेरुलस के पास क्या होता है, ऑल्सो कंटेन्स अ स्मूथ मसल लाइक सेल called as the glomerular mesangial cells. These cells help regulate the blood flow in the glomerulus by contraction and engulf macromolecules that get hung up during filtration. ठीक है, तो mesangial cells भी होती हैं to maintain that blood flow and blood pressure. ठीक है, so this is the podocytes and this is the glomerular capillaries. If you can see how they have bound these capillaries, this is very important structure for kidney to function properly. और filtration membrane कैसे बनती है, ये capillary की membrane है. This is the basement membrane covering the capillary, and these are the foot processes that are formed by the podocytes, which make the filtration slit so as to prevent the flow of larger proteins or important proteins from the capillaries into the filtrate. Okay? So this meshwork of collagen and proteoglycans, fibers that have large spaces for filtration, they also hold strong negative electrical charges. जिसकी वजह से क्या होगा कि प्रोटीन जो है, प्रोटीन जो आ रहा है उस पे भी स्ट्रॉन्ग नेगेटिव इलेक्ट्रिकल चार्ज होता है और इन फूड प्रोसेसेस पे भी स्ट्रॉन्ग नेगेटिव इलेक्ट्रिकल चार्ज होता है, जिसकी वजह से रिपेल करते हैं एक दूसरे को. तो जो लार्जर प्रोटीन है दे कैन नॉट पास थ्रू दिस फिल्ट्रेशन स्लिट्स. ठीक है, तो ये क्या करते हैं, रिस्ट्रिक्ट करते हैं फिल्ट्रेशन ऑफ प्लाज्मा प्रोटीन को. ठीक है, एंड पॉजिटिवली चार्ज जो सब्सटेंस होता है वो फिल्टर हो जाता है इसके थ्रू, क्योंकि नेगेटिव है ना ये. तो चलिए जी, नेक्स्ट थिंग इज कॉम्पोजिशन ऑफ द फिल्ट्रेट. फिल्ट्रेट में क्या क्या होता है, मेटाबोलाइट्स होंगे उसमें, क्योंकि छोटे होते हैं साइज में, दे कैन पास थ्रू दिस आराम से. someone wanted to join, yes, someone wants to join. ठीक है, ओके, उसके बाद है, यस, कॉम्पोजिशन क्या होती है फिल्ट्रेट की. इसके अंदर इलेक्ट्रोलाइट्स होते हैं, मेटाबॉलिक वेस्ट होते हैं, मेटाबोलाइट्स होते हैं, नॉन नेचुरल सब्सटेंसेस, दैट इज व्हाट द थिंग्स दैट आर एक्चुअली फॉर्म्ड फ्रॉम ड्रग्स और पेस्टिसाइड्स और एनी काइंड ऑफ फॉरेन मटेरियल दैट इज नॉट रिक्वायर्ड बाय आवर बॉडी एट ऑल. तो वो सारी चीजें भी इस फिल्ट्रेट में होती हैं. एंड अनदर थिंग दैट इज देयर इज द lower weight proteins and peptides are also there in this filtrate, क्योंकि वो उस slit से आराम से पास कर जाते हैं. ठीक है, तो इसीलिए these things are there in the filtrate which is actually leaving from what, the glomerular capsule. ठीक है, या फिर हम उसको क्या कहते हैं, Bowman's capsule. ठीक है, चलिए जी. अच्छा, फिल्टरेबिलिटी जो होती है, बेसिकली फिल्टरेबिलिटी क्या है, it means that the substance is filtered as freely as water. ठीक है, if the filterability is 1, that means जैसे कि वाटर जो है वो पूरा का पूरा आराम से पास कर जाता है. ठीक है, तो उसकी फिल्टरेबिलिटी 1. ठीक है, सोडियम की भी 1, ग्लूकोज की भी 1 एंड इन्युलिन की भी 1. मतलब these substance can easily pass through this filtration slit and can enter the filtrate. जबकि मायोग्लोबिन और एल्ब्यूमिन जो कि बहुत बड़े प्रोटीन्स हैं, they cannot pass through easily through this slit. So myoglobin की जो filterability है that is 0.75. It is not easy for myoglobin to pass through this filtration slits. And albumin के लिए तो और ही ज्यादा tough है. It is 0.005. So it is very difficult for albumin to pass through this filtration slit and enter the filtrate. Okay, so this is a very important factor that you should know, that what can filter through these filtration slits and enter the filtrate and what cannot. Okay, you guys should know this. Okay, so the glomerular capillary membrane is thicker than the most other capillaries. Okay, but it is also much more porous and therefore fluids filter at a higher rate. ठीक है, ऑल्दो कैपिलरी की जो वॉल है वो थिक है नॉर्मल कैपिलरीज से, लेकिन पोरस भी ये बहुत ज्यादा है, जिसकी वजह से क्या है, फिल्ट्रेशन जो है वो ज्यादा हाई रेट पे होता है. ठीक है. So despite the high filtration rate, the glomerular filtration barrier is selective in determining which molecules will filter based on their size and electrical charge. Table lists the effect of molecular size on filterability. As I told you, जितना बड़ा molecule है उतनी ही difficulty से वो pass on होगा. ठीक है. You should keep this thing in mind. Next is glomerular filtration. The first step in this is फर्स्ट स्टेप इन यूरिन फॉर्मेशन. ठीक है. सो यूरिन फॉर्मेशन में पहला स्टेप क्या है? फिल्ट्रेट का बनना, जो कि कहाँ बन रहा है? बोमेन्स कैप्सूल में. ठीक है. यूरिन फॉर्मेशन begins with the filtration of large amount of fluids through glomerular capillaries into the Bowman's capsule. However, the proteins are not filtered, and if by any chance some protein is filtered, then as I told you, when the fluid is passing through loop of Henle and other proximal and distal convoluted tubule, there are many steps of reabsorption, absorption, and selective absorption and all there in while the fluid is passing through that. So this protein is generally absorbed back in that process, generally. But if there is some kind of a problem, the kidney is not functioning properly, then this protein is actually excreted out, and which is not a very good situation. If protein is being excreted out in the urine, then this situation is a disease situation for that person. Okay. So most salts, organic molecules are freely filtered. Exceptions are few low molecular weight substances like calcium and fatty acid, because they are partially bound to plasma proteins. तो प्लाज्मा प्रोटीन तो वैसे फिल्टर नहीं हो रहे, अगर वो फिल्टर नहीं हो रहे तो फिर ये सब्सटेंस भी इतनी इजीली फिल्टर आउट नहीं होंगे. ठीक है. So this is you should keep in mind. अच्छा, अब एक चीज यहाँ पे आप लोग ध्यान दें, this one arrow one, it shows what? Filtration. This is glomerular capillaries, this is Bowman's capsule, and this arrow one shows what? This is filtration. When the filtration is happening, filtrate is being formed, it is entering into what? The proximal convoluted tubule. ठीक है. अच्छा, ये जो सेकंड एरो इन्होंने मेंशन किया हुआ है, दिस मींस रीअब्जॉर्प्शन. अब मैंने आप लोग को बताया था कि जो लूप ऑफ हेनले है उसके आसपास क्या घूम रहा है? द पेरिट्यूबुलर कैपिलरीज. So what happens? This second arrow is showing the reabsorption into these peritubular capillaries, whichever either it is an ion or it is some important material that is flowing through this filtrate. It is reabsorbed in these peritubular capillaries for the process of reabsorption. And for this, there are many transporters and other things which is required for this reabsorption. Okay. Okay. The three is secretion. This secretion is what? Whatever amount, whatever substance which is not required by the body and is present in the blood, which is not getting filtered through glomerular capsules, can be uh, added to the filtrate by the process of this secretion through this peritubular capillaries network. ठीक है. So this is also very important thing. And fourth is the urinary excretion. When the filtrate has moved through each and every part, all of the process of filtration, reabsorption, and secretion has happened, then at last it will enter the bladder, and through which it will be excreted out. ठीक है. तो ये आप लोगों को पता होना चाहिए कि किस प्रोसेस को हम लोग क्या कहते हैं. ठीक. ठीक है. तो सब्सटेंस है वेस्ट प्रोडक्ट्स लाइक क्रिएटिनिन. ठीक है. तो उनका फिल्ट्रेशन हो जाएगा एंड दे डायरेक्टली एंटर इनटू यूरिन. उनका कोई रीअब्जॉर्प्शन नहीं होता. क्रिएटिनिन का सीधा क्या होता है? एक्सक्रीशन होता है. ठीक. B is the electrolytes. Electrolytes are actually reabsorbed at some point of time in the peritubular capillaries. ठीक है. If you can see, there is this connection. This connection shows what? That it is being reabsorbed into this peritubular capillaries. ठीक. उसके बाद है C, that is amino acid and glucose. They are totally, if you can see, there is not a single percent of glucose or amino acids being excreted out into the urine. They are totally reabsorbed into this peritubular capillaries. ठीक. And last is the drug metabolites. In the drug what will happen? Drug metabolites are also excreted out in the urine completely, and sometimes they are also secreted into this filtrate by the peritubular capillaries, so that they can be removed from the body completely. Okay. So this is what you guys must remember, which thing can be reabsorbed and which can't. ठीक. अच्छा, potassium ions and proton ions या फिर H positive ions are excreted in the urine. Urea, creatinine, uric acid and urates poorly absorbed and excreted in the large amount. ठीक है. पोटेशियम और H पॉजिटिव आयंस ये सब एक्सक्रीट होते हैं यूरिन में, और ये सब भी जो है यूरिया, क्रिएटिनिन, यूरिक एसिड और यूरेट्स, दे आर पुअरली एब्सॉर्ब्ड, दे आर टोटली most of the cases they are totally excreted out in the urine. ठीक है. Foreign substances or drugs are also generally totally excreted out into the urine. Some amount can sometimes stay back due to different reasons, but they are generally excreted out. While the electrolytes like sodium and chloride ions and bicarbonate ions are highly absorbed, they are reabsorbed in the peritubular capillaries. They are not generally excreted out into the urine. ठीक है. तो these are actually very often important for the body to maintain the balance. So yes, they are generally not excreted out. Okay. The nutritional substances like amino acid and glucose are also completely reabsorbed. They are also not excreted out. ठीक है. So this is the amount filtered, amount reabsorbed. Glucose totally reabsorbed. Bicarbonate ions generally totally reabsorbed. Some ions can excrete out depending upon the situation there. Sodium ions are also generally reabsorbed some amount. If you can see 150 is excreted out, otherwise maximum amount is reabsorbed. Chloride ions also maximum amount is reabsorbed. Potassium ions, the amount is some amount is excreted out. Urea also, 50% amount is reabsorbed and 50 is excreted out. It is generally required to maintain the osmotic pressure. So generally यूरिया का कुछ amount जो है body retain कर लेती है. Okay. Creatinine is totally excreted out. कुछ भी amount इसका reabsorb नहीं होता है. It is totally excreted out. ठीक है. Okay, so what is creatinine? Creatinine कब बनता है? Phosphocreatinine, also called as the creatine phosphate, serves as a rapidly mobilizable reserve of high energy phosphates in skeletal muscles and the brain. It anaerobically donate phosphate group to ADP during the first 2 to 7 seconds following intense muscular or neuronal effort. ठीक है. तो जनरली क्या होता है? It is a reservoir of phosphates in the skeletal muscles when there is an intense muscular or neuronal workout is being going on. ठीक है. तो creatinine is chiefly filtered out of the body by kidneys. It is not retained into the body. It is generally filtered out. ठीक है. These are the different pressures that are working under which the filtrate is being produced. So when the blood is flowing, the glomerular hydrostatic pressure, which is working in the direction of the filtrate, it is by the blood flowing through these glomerular capillaries. Plasma colloid osmotic pressure is there. Then the capsular hydrostatic pressure, which is by the capsule wall that is lining these capillaries, the capsule, the Bowman's capsule. It is a double wall structure, and then depending upon the balancing of these different pressures, the net filtration occurs. Okay. So the inward force of plasma colloid osmotic pressure is generally -32, and the outer force, the glomerular hydrostatic pressure is 60, जिसकी वजह से there is a difference in pressure of 18 mm, जिसकी वजह से क्या होता है कि जो नेट फिल्ट्रेशन प्रेशर है दैट इज 10 mm. ठीक है. सो जनरली फिल्ट्रेशन का जो एक्चुअल डिफरेंस है बिटवीन द प्रेशर्स ऑफ जिसकी वजह से फिल्ट्रेट बनता है दैट इज 10 mm. ठीक है. तो इफ देयर इज एन हाई ब्लड प्रेशर, filtration will be higher, but yes, body has mechanisms to prevent that to happen. But if the situation is extreme, then obviously this can lead to some chronic diseases. Otherwise, if the blood pressure of a person is low, then you can yourself understand this, that the net filtration pressure will be low. So the filtrate forming will be low, and this will again cause some kind of problems inside the body. So that's why we should keep our body healthy. ठीक है, ऑनकोटिक प्रेशर हम कर चुके हैं. अच्छा, ऑटो रेगुलेशन, एज आई वाज टेलिंग यू, when there's a high blood pressure in the capillaries of glomerular capillaries, in that situation what happens is this auto regulation of glomerular filtration rate happens, and this actually maintains what, it maintains this pressure and keep the filtrate amount that was flowing through this Bowman's capsule almost constant. ठीक है. So it is an auto regulation. There are two types of auto regulation working. One is myogenic mechanism, and someone is entering, just wait for a moment.
हेलो, आवाज आ रही है क्या?
हाँ जी.
ये स्लाइड, ओके, पहले स्लाइड चल नहीं रही थी, बहुत टाइम बाद चेंज की गई.
अभी मूव कर रही है.
हाँ हाँ, ठीक है.
ओके,
ओके.
you can mute your mic, thank you. Okay, so auto-regulation में it is believed that resistance is controlled primarily at the level of afferent arterioles, ठीक है, rather than efferent arterioles. Why afferent arteriole? Because it has the smooth muscle surrounding this afferent arteriole, which can maintain the, the constriction and the relaxation of this vessel. ठीक है. तो मायोजेनिक मैकेनिज्म में क्या होता है कि इंक्रीज इन रीनल आर्टीरियल प्रेशर जनरली क्या करता है, स्ट्रेच रिसेप्टर्स जो होती हैं इसके वॉल्स के, उनको एक्टिवेट कर देता है, विच रिस्पॉन्ड्स आइदर बाय कॉन्ट्रैक्टिंग और रिलैक्सिंग. ठीक है, विच मेंटेन्स द ऑटो रेगुलेशन इन दिस ब्लड फ्लो. ठीक है, तो दिस इज मायोजेनिक मैकेनिज्म. ट्यूबुलोग्लोमेरुलर फीडबैक में क्या होता है, there is this macular densa. If you guys remember when I was explaining you guys the structure of nephron, I told you there is this macular densa. Macular densa is what, it is the group of cells, ठीक है, that is present in the distal convoluted tubule. तो ये क्या होता है, distal convoluted tubule when it is bent near to this glomerulus or this Bowman's capsule, then this macular denser cells actually sense what, the concentration of sodium and chloride ions. ठीक है. तो this sense of calcium and chloride, sorry, sodium and chloride ions actually helps this macular denser to understand that whether there is a requirement of water regulation in the kidney or not. So let's read what this says. The macular densa is a collection of densely packed epithelial cells at the junction of thick ascending limb and distal convoluted tubule. As the thick ascending limb ascends through the renal cortex, it encounters its own glomerulus. As I told you, it is a little bit bent near the glomerulus, bringing the macula densa to rest at the angle between the afferent and efferent arterioles. ठीक है. So macula densa's position enables it to rapidly alter glomerular resistance in response to the changes in the flow rate. जो भी flow rate है उसके according to the sense what they are getting, उसके basis पे ये क्या करता है, flow rate को change करने में help करता है through the distal nephron. ठीक है. So in response to the increased flow of tubular fluid in the thick ascending limb, increased sodium chloride concentration at macula densa, या फिर इंक्रीज सोडियम क्लोराइड कॉन्सेंट्रेशन होगी एट मैक्यूला डेंसा. तो क्या होगा, elevated filtration at the glomerulus or reduced reabsorption of the sodium and water by proximal convoluted tubule causes this tubular fluid at the macula densa to have a higher concentration of sodium and chloride. ठीक है, ये देखिए. तो यहाँ पे फिर NKCC2, if you guys remember, sir also emphasized on this NKCC2 co-transporters. So these co-transporters will come into action. Why they will come into action? Because at the distal end of these macular denser cells, where the blood flow is there in the peritubular capillaries, so what will happen? This sodium potassium transporters will come into action. Yes. If you can see this sodium potassium transporter, sodium potassium ATPase will come into action, and the sodium will start moving in the direction of this blood flow. This will lead to this macular denser cells to absorb this sodium and chloride ions with the help of NKCC2 from the interstitial fluid, and which will further lead to the let's see what this is, cellular mechanisms. Increase in the delivery of sodium and chloride to macula densa causes vasoconstriction of the afferent arteriole of the same nephron. Afferent arteriole is the arteriole through which blood is flowing inside the nephron. ठीक है, efferent से जाता है, afferent से आता है. ठीक है, so that is tubuloglomerular feedback. So an increase in GFR elevates the sodium chloride in the tubule fluid at the macula densa. This in turn enhances the uptake of sodium and chloride ion across the apical membrane of the macula densa via NKCC2 co-transporters. ठीक है, सिम्पोर्टर्स भी इनको हमने कह दिया, co-transporters भी, which leads to an increase in adenosine formation. ATP and ADP will start moving outside, and उससे क्या होगा कि जो ADP है और ATP है, they will start producing adenosine, which will further sensed by what, the A1 receptors या P2X इसलिए कहा हमने क्योंकि प्यूरिनर्जिक रिसेप्टर है. So these receptors will sense this adenosine and will lead to the vasoconstriction. ठीक है, so ATP binds to this P2X purinergic receptor and adenosine binds to adenosine A1 receptor in the plasma membrane of smooth muscle cells that are lining what, afferent arteriole. ठीक है, मैंने बताया था ना एफरेंट आर्टीरियल के आसपास स्मूथ मसल्स का अमाउंट बहुत ज्यादा है. एंड फिर क्या होगा कि सराउंडिंग द एफरेंट आर्टीरियल, both of which will increase the intracellular calcium ion, and we all know calcium ion is important for the muscles to contract. ठीक है, सो राइज होगा कैल्शियम आयन का, स्मूथ मसल्स कॉन्ट्रैक्ट करेंगी, स्मूथ मसल्स कॉन्ट्रैक्ट करेंगी तो इट विल लीड टू द वेसो कॉन्स्ट्रिक्शन. तो ब्लड फ्लो जो है वो थोड़ा हिंडर होगा, और जो ग्लोमेरुलर फिल्ट्रेशन रेट है जो हाई हो गया था, दैट विल बी लेसन डाउन. ठीक है, so this is what macular denser does. It is very important for the auto regulation of the kidneys. ठीक है, next is erythropoietin. As I told you, erythropoietin are what, these are very important for the formation of RBCs. ठीक है, RBCs के लिए क्या होता है, और ये होते कहाँ पे, erythropoietin किडनी में बनता कहाँ पे है, तो if you can see this is formed in the what, in the cortex, renal cortex. ठीक है, renal cortex में जो है erythropoietin बनता है, and it is very important for the formation of RBCs. and ये किसके रिस्पॉन्स में, लो ऑक्सीजन की सिचुएशन में जो है ये एक्टिवेट होता है, और इट runs through the blood and reaches the bone marrows where the RBCs are formed. ठीक है, तो bone marrow में बनती है RBCs, so it must reach the bone marrow to increase the production of RBCs. So the principal factor that stimulates RBC production in low oxygen condition is erythropoietin, a glycoprotein with molecular weight of 34,000. ठीक है, या फिर 34 केडा बोल सकते हो आप इसको, 34,000 इन्होंने डेल्टन्स लिखा होगा. So it is also called as a hematopoietin or hemopoietin. ठीक है, erythropoietin, as I told you, this erythro word is used for red blood cells. ठीक है, so in the absence of this substance, hypoxia has no stimulatory effect on RBC production. अगर इराइथ्रोपोइटिन ही नहीं होगा तो आरबीसी प्रोडक्शन इंक्रीज नहीं होगी, तो चाहे कितनी हाइपोक्सिक कंडीशन हो, ऑक्सीजन की कमी हो बॉडी में, आरबीसी प्रोडक्शन विल नॉट एलिवेट. ठीक है, तो इराइथ्रोपोइटिन इज वेरी इंपॉर्टेंट फॉर आरबीसी प्रोडक्शन. ठीक है, सो इट इज प्रोड्यूस्ड बाय द इंटरस्टिशियल फाइब्रोब्लास्ट इन द रीनल कॉर्टेक्स. Fibroblast is the type of cells present in the renal cortex. So these interstitial fibroblasts actually makes what, erythropoietin, and in the close association with the peritubular capillaries and in the tubular epithelial cells. If both the kidneys are removed or they are destroyed by some renal disease, anemic condition is generated. ठीक है, about 10% of erythropoietin is produced in the liver, but that is not much sufficient. It can work to some extent, but yes, the production of erythropoietin by the kidneys is very 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 important. ठीक है, तो दिस एज आई टोल्ड यू कि टिश्यू ऑक्सीजनेशन जैसे ही डिक्रीज होती है इट विल लीड टू द सिक्रीशन ऑफ what erythropoietin production, and it will lead to the it will stimulate the hematopoietic stem cells to form the red blood cells. And when this amount of red blood cells increases in the blood, tissue oxygenation is proper, तो फिर क्या होगा कि this will decrease the production, kidneys will be activated and they want kidneys will get the stimulation and they won't produce this erythropoietin anymore, and the blood flow will be maintained as it was. ठीक, this is important, and you can see erythropoietin can work on all of these cells, which will further lead to different kind of the blood cells production. ठीक है, okay, so this is something you should keep in mind कि proximal convoluted tubule में जो potassium है, sodium है, amino acids हैं, these are reabsorbed. Glucose, water, sodium, and potassium. These are reabsorbed in the proximal convoluted tubules. ठीक है, जब लूप ऑफ हेनले की बारी आती है तो इन लूप ऑफ हेनले ऑल्सो सोडियम इज रीएब्सॉर्ब्ड. ठीक है, उसके बाद potassium and H positive ions are secreted into the filtrate in distal convoluted tubule. Again, there are many kind of transporters, symporters, antiporters. Everything is involved in this secretion and reabsorption, and this is just to maintain the ionic balance inside the blood. So some amount of ions are uh, secreted into the distal convoluted tubule also. And then in the collecting duct, when there's the situation come when the aldosterone or antidiuretic hormone comes into action, then the water is also reabsorbed from the collecting duct. Otherwise, if there is excess amount of water available in the body, then this excess amount of water is excreted out in the urine. ठीक है, so as the glomerular filtrate enters the renal tubule, it flows sequentially through the successive parts of the tubule: the proximal tubule, the loop of Henle, the distal tubule, the collecting tubule, and finally the collecting duct before it is excreted as the urine. ठीक है, and if you can see, these proximal convoluted tubules have these microvilli present. These microvilli are for the reabsorption. If you guys remember, or if you guys know already, then these microvilli are also present in the small intestine to increase the absorption area. ठीक है, so these are also present in this proximal convoluted tubule, which is not there in the distal convoluted tubule. ठीक है, so guys must remember this. So this is all what the reabsorption. Acha, the reabsorption can occur through two routes: a paracellular path, a transcellular path. The paracellular path generally इससे जो है water या फिर बहुत small ions can pass through it, not much but yes. And the transcellular path generally involves the usage of many kind of transporters, channels, symporters, antiporters, and different systems. So there are two paths through which the amount can things can be reabsorbed and can be brought back to peritubular capillaries. ठीक है, so this is what the proximal tubules, as I told you, they have microvilli, so they have a high capacity of active and passive reabsorption. So maximum reabsorption अगर हम कहें कहीं पे हो सकता है किडनी में, तो that is at the proximal tubules level for ions and many things like amino acids and metabolic things. ठीक है, active transport requires ATP, secondary active transporters are involved, primary transporters are involved for this active transport. So this is something you guys must have known. As I told you कि यहाँ पे क्या है, सोडियम ग्लूकोज सिम्पोर्टर्स भी होते हैं, which is actually for the reabsorption of glucose, amino acids, and some ions and vitamins and things which are important for the body, which shouldn't be excreted out in the filtrate. And this sodium ion balance is generally maintained by the sodium potassium pump or sodium potassium ATPase that is present at the basolateral level of these tubular cells. ठीक है, so this is all what is saying. अच्छा, GLUT is there. GLUT2 is basically for the transport of glucose from these tubular cells to the blood. So GLUT is a transporter for glucose. ठीक है, now SGLT family. This is what the sodium dependent glucose transporters, as I told you. So these are also known as the co-transporter. When the glucose is being transported into the tubular cell through this from this filtrate, then it must transport sodium also from that filtrate to the tubular cells, because this is a co-transporter. This works this way only. One thing that you should always keep in mind, that this SGLT family protein, this transporter, is a very large protein. It is a 14 pass protein. If you guys remember, in the last lecture or last lecture, we have discussed the 7 pass transmembranal protein. Which was that? It was a G protein. That was also a larger protein, and it is 14 pass transmembrane protein. So you guys can imagine how large this protein is. So this is a co-transporter which transport glucose and sodium ion inside the tubular cells, from where they are transported back to the blood through the sodium potassium ATPase channel and through GLUT2, which is a transporter of glucose. Okay? And there are different types of SGLT family co-transporters, and locations for them is different in different organs. ठीक है? So this is something. अच्छा हाँ, लार्जर प्रोटीन्स को रीअब्सॉर्ब करने के लिए जो सेल्स होती हैं पीसीटी की, दैट इज प्रॉक्सिमल कॉन्वोल्यूटेड ट्यूब्यूल, दे कैन नॉट ट्रांसपोर्ट that through any channel or something. So there must be some other system to reabsorb these larger proteins, or things which must be uh, reabsorbed in the tubule cells. So for that, what mechanism they use? This is pinocytosis. Pinocytosis is a mechanism that is used by the tubular cells to reabsorb the larger proteins. So this is also you should keep in mind. This is a transport maximum. This is what this is if the amount of glucose that is entering, that is in your blood, and that is filtered into the filtrate, then there is a transport maximum situation there, which is a situation when this is the maximum amount of glucose that can be reabsorbed from this filtrate. Rest all of the amount which cannot be reabsorbed, when the transporters are saturated, in that situation the excess amount of glucose is excreted out, and the higher amount of glucose in the in the urine is not a good situation. We all know this is a diabetic situation, and this is a situation which we should be concerned about if there is some person who has this situation. So transport maximum is that condition in which the maximum reabsorption, or we can say the maximum transport of that substance from filtrate to tubular cells and then to the interstitial space or the peritubular capillaries can happen. So for glucose it is 375 mg per minute. इससे ज़्यादा अगर glucose is there in the filtrate, then that can't be reabsorbed. ठीक है? Phosphate, and for different substances this is different values. ठीक है? Passive water reabsorption occurs by the osmosis, coupled with sodium reabsorption also, and also a large part of osmotic flow of waters occur through the so-called tight junctions. There are tight junctions present between the tubular cells, so through which, as I told you, there is a paracellular way also for the transport of some things through these tubular cells, and water is one of them. Another thing that can pass through is some amount of chloride ions, sodium ions, potassium, and these ions can pass through it. अच्छा, bicarbonate ion जो filtrate में present होता है, that must also be reabsorbed. Maximum amount of that should be reabsorbed, for which what happens is carbonic anhydrase come into action, which actually transports this what bicarbonate ion back into the tubular cells, and from tubular cells into the blood. It is important to maintain the pH of the blood. So it must be reabsorbed from the filtrate and it should be sent back into the blood. Okay? So this was that. Now let's come to the assignment questions. I hope everyone was able to do so. If someone had any problem, they can ask me now. Otherwise, I'll just let you go through it once. And then we'll practice some questions, and apart from that, if anyone wants to ask some question, they can ask. Okay, So yes, guys, what is one? Everyone can unmute their mic and answer. And I would like if everyone will participate, this will help you guys only. What is this thing that is marked as one? Anyone? Okay, I'll tell you, this is afferent.
Afferent lymphatic.
Bilkul, bilkul, very good. It is an afferent lymphatic vessel. What is this thing that is marked as 2? Which circulation is there? If you can understand, this is an heart. So which circulation will be it be?
Systematic circulation.
Yes, it's systemic circulation. Third is, this is what? इंटरस्टिशियम के पास में कौन सी कैपिलरीज होंगी जो एक्सेस इंटरस्टिशियल फ्लूइड को कलेक्ट करती हैं? These are lymphatic capillaries, okay? And this fourth is a lymph node. ठीक? Let's move to the next thing. This is what? Everyone, what is this? The one which is marked as 1. What is this?
Ureter.
What is this thing which is marked as 1, everyone? It is ureter. This is the tube which is taking filtrate from kidney to urinary bladder. Okay. And the thing which is marked as 2 is जहाँ पे कौन सा हार्मोन बनता है? इराइथ्रोपोइटिन. What it is?
Renal cortex.
Renal cortex. Very good. The third is, which is marked as third is?
Renal pelvis.
Renal pelvis, right? And which is marked as fourth is what? किस चीज का extended enlarged structure दिखाया है ये?
Nephron.
Nephron, right? Very right. Next question. What is this thing which is marked as 1?
Glomerulus.
Glomerulus, glomerular capillaries, which is marked as 2?
Afferent arteriole.
Efferent arteriole. Third is
Efferent arteriole.
efferent arteriole, because they are leaving the glomerular capsule. ठीक है? Fourth is, अगर
Loop
आप
of
लोग,
Henry.
loop of Henle. Yes. ठीक है जी. Now, which is marked as 1 is
Podocytes.
podocyte pedicels. Yes, these are the foots of the podocytes. 2 is
Basement membrane.
basement membrane, but यहाँ हम लोग इसको filtration membrane भी कह सकते हैं,
Filtration membrane.
because filtrate is coming out through this membrane only. Okay. Third is
Endothelium.
endothelium cells, and fourth is, you can guess it from the
RBC.
structure only, RBC. Yes. Okay. Which is this thing which is marked as 1? RBCs. These are red blood cells. Which is marked as 2 is tissue oxygenation. Okay. And kidney से क्या बनता है? तो जो third, which is marked as third is, what is that? Which hormone?
Erythropoietin.
Erythropoietin. Yes, it is erythropoietin. Okay. So now, as we always do in our session, we'll proceed with some other questions that I have brought for you guys. So let's start with question number 1. Which of the following is not a function of the kidney? बताइए मुझे.
Option B.
Which is not is the function of kidney? And I would like if you guys will write this answer in a chat box so that everyone can participate. Are the slides visible to you guys? Okay, so your answer is B. बाकी लोग? although the people are less, but obviously you all can answer. So yes, the answer is B. The production of bile is not the function of kidney. Okay. Question number 2. Which of the following conditions leads to a decrease in RBC production? किस सिचुएशन में आरबीसी प्रोडक्शन जो है वो डिक्रीज हो जाएगा? वी हैव डिस्कस दिस टुडे. Yes, in case of the chronic kidney disease. In that situation, the RBC production will decrease. Okay. Next question is, which enzyme is crucial in the conversion of CO2 into bicarbonates ion in RBCs? बाकी लोग कोई आंसर नहीं करना चाहेगा? There are 2-3 people more here. Lakshmi. Yes, Rajatha. Lakshmi. No problem at all. Okay. So yes, the answer is answer B. Yes, yes. It is carbonic anhydrase or carbonic anhydrase only. ठीक है? Okay, let's move to the next question. What is the basic functional unit of kidney? Basic functional unit of kidney. Answer to this question. Yes, it is A, that is nephron. ठीक है? Which part of nephron is responsible for ultrafiltration? Ultra what is used for pressure? राजा था, ये आंसर आपने जो लिखा है ये दिस इज फॉर क्वेश्चन ए और क्वेश्चन 4 और 5. ओके, फॉर 5 यू हैव रिटन बी. ओके, चलिए जी. तो यस, इट्स आंसर इज बी. ठीक है, इट इज द ग्लोमेरुलस ओनली. नेक्स्ट क्वेश्चन, व्हाट इज द प्राइमरी फंक्शन ऑफ लूप ऑफ हेनले? प्राइमरी फंक्शन क्या होता है लूप ऑफ हेनले का? एवरीवन आंसर. ओके, तो आप लोग कह रहे हैं इट्स आंसर इज बी. So yes, it is correct. Reabsorption of sodium and water as much as possible. ठीक है? चलिए, next set of question. Which hormone increases water reabsorption in the kidney? बताइए. Yes, it is antidiuretic hormone ADH. ठीक है? Next question is, what is the normal glomerular filtration rate in a healthy adult? अगर आप लोगों ने लेक्चर ढंग से सुना था सर का तो यू गाइस मस्ट रिमेम्बर दिस. क्वेश्चन नंबर 8 का आंसर. Yes, it's B. 90 to 120 ml per minute. Which nitrogenous waste is excreted primarily by the kidneys? Which nitrogenous waste is excreted primarily by the kidneys? Again, yes, the answer is B, urea. It is urea which is excreted by the kidneys. ठीक है? Okay. What is the main driving force for glomerular filtration? Main driving force for filtration बताइए. It is hydrostatic pressure. Yes, it is hydrostatic pressure. What is the primary function of lymphatic system? Again, the answer is B. Yes, immune response and fluid balance. Next is lymphatic vessels transport lymph to which major circulatory system? ये हम लोग आज डिस्कस कर रहे थे. The answer is A. Yes, it is venous system. ठीक है? So these are some questions. There are still some 8 or 9 questions left that I am leaving for you guys. You can find it in the notes section that I'll be uploading. आप लोग वहाँ सॉल्व कर लीजिए. किसी को कोई प्रॉब्लम है, you can ask me in the next lecture. Apart from that, if someone has any confusion, if someone wants to ask me anything, they can ask. Any other confusion if you guys have, you want to ask something, you want to include anything in the lectures, you want something else to be done in the lectures, like something else to be discussed in the lectures, you can tell me. More elaborated topics discussion, I can do that also. You can ask me anything now from the lecture, obviously. Any questions, everyone? Yes, I'll be uploading this PPT. I'll be uploading this PPT on the link that I have provided, that is provided to you guys. So you can get it from there. Otherwise, you must have gotten the lecture material that was uploaded by sir. Apart from that, you will get this PPT also.
Thank you, ma'am.
You're welcome. Any other thing anyone wants to ask? If no, then we can conclude this meeting. You guys can leave the meeting and then I'll be ending the session. If no one wants to ask anything, they can leave the meeting.
Thank you, ma'am.
You're welcome. You're welcome, Suresh Balaji.
Thank
चलिए जी. Let's